Welcome to the ATiO Podcast, the ultimate gateway to first-hand insights from world-renowned experts in the realm of advanced therapies and orthopedics, unraveling the latest advancements and perspectives. Join us as we navigate this intricate landscape right here on the ATiO Podcast.
We are honored to welcome Jonathan Steckbeck, founder and CEO of Peptilogics, alongside Nicholas Pachuda, COO of Portfolio Development. Peptilogics is a clinical stage biotech company, combining computational design, deep learning, and purpose-built supercomputing to create solutions for severe infections in orthopaedics and beyond. Their lead candidate, PLG-0206, is engineered to treat periprosthetic joint infection, an area with immense unmet clinical need. Dr. Pachuda brings nearly 3 decades of clinical and industry experience, including leadership roles in R&D, clinical development, business development, and commercial strategy, with a foundation in orthopedic surgery and trauma. Jonathan has guided the company's mission to harness AI-assisted peptide discovery to tackle infections and biofilm challenges. Just recently, Peptilogics won the 2025 ATiO Emerging Company Pitch Contest, recognized for their innovation in infection treatment and prevention through peptide therapeutics. In today's episode, we will explore how their award-winning technology came to be, the next steps following this important recognition, and their mission for the future of musculoskeletal therapies. Their winning presentation was titled, "Changing the Paradigm of Infection Treatment and Prevention." Jonathan, Nick, it's a pleasure to welcome you to the podcast.
Thanks for having us.
Absolutely. Great to be here.
Could you briefly introduce yourself to the audience and share your professional journey so far?
Yeah, I'm Jonathan Steckbeck, the founder and CEO of Peptilogics. I started the company in 2013 to create and advance new therapeutics, starting in the med device infection space. And now we've got a pipeline of products that are both treating and preventing medical device infections.
Yep. And this is Nick Pachuda, Chief Operating Officer of Peptilogics for the last 5 years. Surgeon by training, practiced for 10 years after completing an AO trauma fellowship. Worked for 3 large multinationals, including Arthrex, Synthes, and J&J. Been lucky enough to be part of 5 exits, 3 as a leadership team, 2 as a board member, and met Jonathan in 2018 when I was leading external innovation for J&J. We had a focus on anti-infection. And when Jonathan was working on the Peptilogics PLG-0206 for the potential treatment and prevention of medical device infections, I knew it was a really strong fit for what we were looking at. And I was lucky enough to be able to join the company in 2020 and help Jonathan build team and move it to clinical trials.
So what inspired the founding of Peptilogics and what are the core objectives?
Yeah, so I can handle the first part. So, I started Peptilogics really, like I said, around the idea of being able to treat kind of originally drug-resistant bacterial infections. And so our technology is an engineered antimicrobial peptide. And we had done a lot of work to overcome some of the challenges in that molecular class. So the class had been around about 40 years and always had clinical development challenges around toxin safety. And so we designed the molecule with 2 goals in mind. 1, to be more active. 1, to make it non-toxic and safe. And, over time we were developing it initially because I think of it as a systemically administered antibiotic for bacterial infections against drug-resistant bacterial infections. As the commercial challenges became apparent in that space, we had some really interesting data from an orthopedic surgeon collaborator who was using it in his models of treating periprosthetic joint infection. And he called me one day, had really great results. We took a pretty hard look at the business case for kind of treating, again, PJI kind of as an indicative for device-related infections, and saw that the opportunity there for kind of commercial success was different than we would have expected as an antibiotic company. And so at the end of 2017, early 2018, we made a wholesale switch, and now we're only focused on looking at, like I said in my kind of intro, treating and preventing medical device related infections.
Yeah, I mean, from a clinical perspective, I've known for years that treating and preventing infections are really challenging. They become surgical problems all the time. It's not something that's really managed with systemic antibiotics. So these are, urgent, if not emergent issues quite often that have to be dealt with on the same day, typically. And there's no great option. You're treating it with a bit of a belt and suspenders, a shotgun approach, trying everything you can. And there's been really no effective treatment. So these, you find your patients having to undergo multiple surgeries to resolve the issue. And what we were really looking for, back in my days at J&J was something that could treat infections in the operating room in a surgical setting, but also required data and a claim that could enable a product to be globally scalable and also be reimbursed, to really make it a viable, not only a clinical case, but a business case. And we looked at a number of technologies, coatings, antimicrobials, antiseptics, et cetera. And what we really settled on is that the solution needed to be independent of the device. And that's at the same time that I was sort of learning that the business case around being implant agnostic or implant independent, a freestanding product, is when I met Jonathan. So when I saw a technology that could be used in the operating room that worked fast, broad spectrum, and could eradicate potentially infection and was safe on human tissue, that's what I was looking for, and that's why I joined the company. And we're starting with periprosthetic joint infection, but the interest was in the platform technology and the broader applicability around treatment and prevention. So that's absolutely why I joined, because I see the potential to address maybe one of the biggest problems in not just orthopedics, but in the medical device world, is trying to eradicate these infections.
Impressive. Before we dive deeper into the technology and also your lead product, Jonathan, you mentioned commercial challenges. What do you mean with that?
Yeah, so kind of a systemically delivered antibiotic, whether it's oral or intravenously delivered, what we have seen over the last decade to 15 years is that when a new antibiotic comes on the market, the reimbursement, so there's a reimbursement challenge. And, what the challenge ends up being is that, a hospital is reimbursed for the average case through the bundled payment. And a new antibiotic, as kind of any other drug, you need to be able to charge a reasonable price in order to be able to recoup your investment. And, we all run businesses. And what the main challenge for antibiotics is that the bundled payment system doesn't allow for essentially payment for innovation. And because antibiotic resistance isn't an enormous problem, which is good for all of us, but because the majority of cases aren't treating a drug-resistant bacteria, and some of the stewardship concerns. And what that means is infectious disease doctors don't by default want to use the newest drugs because they want to make sure that they're not promoting resistance. So you have this confluence of payment kind of characteristics as well as use characteristics in the antibiotic space that just leads to very little, kind of use and uptake when a drug is launched. We expect that to be different in, again, starting in PJI, but in the device-related infections, because the drugs, existing antibiotics don't do a good job of clearing the infection off of the device, and it requires surgical intervention.
Speaking of PGIs, you mentioned your lead product, PLG0206. Can you explain the science and technology behind your solution in simple terms, also for the broader medical audience?
Yeah, so the way 206, PLG0206 works, and I'm just going to call it 206 because it's easier to, it just kind of flows a little bit better, is that, so as I mentioned, it's an antimicrobial peptide. And the main difference in how it works from traditional antibiotics is that it works from the outside in. And what I mean by that is that it interacts with the bacterial membrane, that outer kind of coating, right? The thing that separates the outside from the inside. And it binds to the membrane, kind of inserts itself in the membrane, and then it changes the mechanical properties. And what that means is that kind of effectively where the drug is sitting on the membrane, it creates kind of thick, stiff areas of membrane that sit right next to thin, very fluid, flexible areas of the membrane. And you get a kind of a size mismatch. And the ultimate kind of impact of that is it lets water leak across the membrane freely. And so that's something that bacteria and all cells typically very tightly control in order to be able to make energy. Because it kind of just opens that up, the bacteria can no longer make energy and they rapidly die. Now, this is important for biofilm-based infections like those that occur on medical devices, because biofilms end up being structured communities of bacteria that kind of exist in two different metabolic states or two different growth states. And so the majority of the population is actively growing, but a small percent, think 5% or so, are just laying there dormant. And what happens, or the consequence of that, is when you treat a biofilm with a typical antibiotic, or kind of something that you would normally think of as an antibiotic, those mostly work on preventing or inhibiting growth processes. So they're pretty effective over time at eliminating that 95% of the bacteria that are actively growing. But that 5% that's just dormant, they can't do anything about. And so then when you remove the treatment, the infection, that 5% of bacteria rapidly grows back. And that's what causes the high recurrence rates that we see in these types of infections. 206, because it works on the membrane, is equally effective against both those very actively growing, but more importantly, against that 5% of dormant bacteria. And so we've shown, in the lab, in animals, and in clinical studies, where we're able to effectively eliminate all of the biofilm bacteria very rapidly, as Nick said. And that's why we see such promise for this technology.
So you also mentioned that the usage of your product is not as prone to create resistance in bacteria. Is it because of the mechanism of action? Is it because it's more effective in eradicating the biofilm antibacteria? Could you clarify that?
Yeah, it mostly comes down to mechanism of action. Because it's targeting the membrane, which is very evolutionarily conserved, let's say. It's the one thing that separates bacteria from kind of mammalian or human cells, the content of that membrane being different. It's very difficult for bacteria to evolve resistance. And so we've done, a ton of testing to show that, under clinical conditions, we don't expect there to be the evolution of resistance. Because we're administering this as a very high concentration, single dose. You can essentially think of it as, be careful how I say this. You can think of it as kind of like setting off a nuclear bomb and just eliminating all of the bacteria. And there's not really a chance for them to survive.
So because of that mechanism, what has that enabled you to do, especially in the surgical setting? Because if you think about the challenges of orthopedic infection, and you can go to the American Academy of Orthopaedic Surgeons and you'll hear surgeons on the podium saying that this is the most catastrophic problem. The biggest unmet need in orthopedics is to be able to address infection. And the treatment today is not that dissimilar than it was 50 years ago or 100 years ago. When something gets infected, you've got to do surgery, remove the infection, remove the necrotic tissue, and very often the gold standard is removing the implant, which is the main reason you did the surgery in the first place, is to replace a joint or to stabilize a fracture or to fuse a spine. And when you have to take that hardware out, you not only have an infection problem, you now have, a bone stability, a mobility problem for that patient. So these patients undergo multiple operations. They have mobility issues. And unfortunately, in a joint replacement patient, these patients are typically older. They have multiple comorbidities. So when you add the impact of lack of mobility on a patient that's got, say, a high BMI and diabetes and hypertension and a heart problem, it compounds the issue. And you end up with a mortality rate at 5 years that's about 25%, which is higher than 4 of the top 5 cancers in the world. So surgeons have been treating these surgically with repeat operations, repeat hospitalizations, and partly because systemic antibiotics can't get to the site of the problem. You don't have blood supply to metal and plastic and ceramic. You don't have great blood supply to damaged bone, and certainly, to the biofilm, so that it sets up sort of a perfect nidus for infection and reinfection. So the ability to be able to deliver a highly efficacious agent to the site of the problem during an operation, during a normal surgical workflow, have it eradicate both gram-positive and gram-negative bacteria and the biofilm, and try to do that in one setting. The patient can retain their implant and keep moving is really the critical end result. Is the unique mechanism gives you a unique opportunity in the operating room for a surgeon to minimize the number of procedures, minimize the number of surgeries and hospitalizations, and therefore keep that patient moving, keep them away from some of those more impactful medical side effects of not being able to be mobile.
Coming back to the analogy of Jonathan, and also more practically, how do we apply the product? I remember that in the title of your pitch, you were also speaking about prevention.
I'll let Nick's a surgeon, so I'll let him talk about that one.
Yeah, I think these surgeries, these are still surgical problems, not only because you have bacteria and biofilm on a medical device and in and around the joint, but you have necrotic tissue. When there's infection, you've got to remove the tissue. So that you have to do a thorough debridement, and you see that there's research that's done that the more thorough the debridement, the longer that debridement goes, the more chance of preventing reinfection. So you need to do an aggressive debridement, serial irrigation with our product being used as the last step in the irrigation. It's aspirated from the wound, but it is not washed out or irrigated. It is the last irrigation the patient sees before the wound is closed. So it works in the standard surgical workflow of any surgeon, whether it's a joint replacement surgeon, spine, trauma, et cetera. So it is simple to use, and we're not disrupting that first surgery. What we're trying to do is prevent the second surgery and the third surgery that those patients see. So that's where we see the best indication for treatment of established infections. But the implications, and we hear that from surgeons around the world as well, if you can use 206 to treat these catastrophic problems, how can I prevent them? How can I go upstream and not have this happen at all? So that's why we're working on our second program, which is to be able to deliver 206 over a 2 to 4 week period and put them on the primary index surgery of the elective joint replacement or fracture surgery or spine surgery down the road, where the product is used as a preventative agent, a prophylactic used locally at the end of the surgery, as opposed to a preoperative systemic IV antibiotic that typically only covers about 60% of the bacteria that contaminate these devices. So our lead project for treatment is an irrigation at the end of surgery. Our prevention product is going to be something that is a controlled release that releases antibiotic at a therapeutic level into the wound for hopefully up to 4 weeks.
And what kind of bacteria does 206 cover?
Yeah, 206 is, as Nick mentioned, I think we said a couple of times, is broad spectrum. So it covers all of the clinically relevant gram positives and gram negatives. So you can think, MRSA, Staph aureus, the coagulase negative staphs, Enterococcus, kind of as the big ones. And then on the gram negative side, Pseudomonas, E. coli, et cetera. So it's truly broad spectrum.
Impressive. To summarize, so 206, the primary application of your product, or rather the clinical setting, is currently only for PGI?
So our first trial is in PJI. So we did a phase 1B trial in knee PJI patients. We're starting a pivotal phase 2/3 trial in knee PJI patients. But really we think about, if you take a step back and say, okay, what's the cause of the problem? It's bacteria, biofilm on device materials, right? Metal, plastic, cement, et cetera. Really what we look at, the clinical data that we're getting in treating PJI, as really kind of our position is that it establishes kind of clinical proof of mechanism in being able to clear biofilm related bacteria from device, from device-related infections, and it should be applicable across other diseases as well, or other anatomical sites.
Yeah, because if you think about what triggers many medical device infections, it's typically the skin flora that this hardware is exposed to during the time of the procedure. And that skin flora obviously isn't different, whether you're having a joint replacement or a fracture surgery or a spine surgery or a pacemaker, it doesn't matter. So our goal, obviously, we're starting with a knee PJI DARE trial, but we fully intend to continue to accumulate data and work with the regulatory bodies to ensure that over time, we will broaden the indications and the reimbursement for this product across all orthopedic and medical device indications. Because, as Jonathan said, the pathology and the mechanism of these infections is similar across the board. So we see broader applicability of the platform.
And just to be clear, so it is more effective than traditional antibiotic therapies and also single dose?
Yeah, the irrigation product is administered as a single dose at the end of the surgical procedure, as Nick said. And yeah, we have pretty significant amount of data, head-to-head comparisons of 206 versus a plethora of traditional antibiotics. And what we see invariably across the board is that in biofilm-based infections, 206 clears the bacteria when traditional antibiotics does not.
Yeah, and clinically, the implication of the current standard of care of surgery and systemic antibiotics and putting various antiseptics or antimicrobials off the shelf, off-label into the wound is that, with a DARE where you're irrigating and debriding and retaining the hardware, the reinfection rate is still around 50% at 1 year. And the gold standard is a 2-stage where you're taking the hardware out, hoping to resolve the infection, putting hardware in down the road. That reinfection rate has been between 15 and 25%. So these are still catastrophic problems with surgeons trying both surgery, local antiseptics, and systemic antibiotics, sometimes for very long term. And these patients still have recurrent infections. So that's the current standard of care. There is no product yet that's come through the FDA as an approved therapeutic to treat medical device infections or prevent medical device infections. So to be clear, we're starting with a PJI knee DARE trial, a randomized controlled trial against a placebo control. And we're doing that as a superiority trial to prove that, we are superior to the standard of care that physicians are doing today, because we're looking for this to be a complete paradigm shift in treatment once the product is commercially available.
Did you already apply 206 in other joints?
We have not yet.
Jonathan, maybe you can talk a little bit about the preclinical, the work we've done, the rabbit and the ex vivo, because there are some implications there.
Yeah, so we did. So as part of our early testing, once we got outside the lab, we used some larger animal models to test its efficacy in treating PJI and as a surgically administered product. So there, a challenge, of course, is that for a lot of animals, you don't have arthroplasty kits available. So we had to kind of make up our own model. And so there, you take a rabbit knee and you drill a hole in the tibia, insert a pin, and then once you surgically close them, you inject bacteria into the knee, let it incubate for a few days to kind of form that biofilm-related infection. And then we would go through and we would treat the same way that you would treat a human, right, with a DARE. So open the joint, kind of do a debridement, irrigate with 206, and then close it up. And we saw there, 60% of the animals survived there. The implications is, again, not having an arthroplasty kit, you're drilling a channel into the bone. And so what you're really setting up is an osteomyelitis often. And so the fact that we got really good results there with, animals surviving 30 days with no antibiotics showed that 206 was able to clear those things off. And so that was kind of a good in vivo experiment. At the same time, we were also testing, as Nick mentioned, we did an ex vivo study. So patients undergoing a 2-stage exchange, whether it was knees or hips, that had the infected joint removed. We would take that and then treat it ex vivo to kind of get an understanding of actual kind of clinical bacterial burdens, but also kind of the complex environment that exists between materials, kind of bodily fluids, tissue, et cetera. And there, I think we tested 17 patients. 16 of the 17, we were able to reduce below a detectable limit. So effectively sterilizing those infected implants in a clinically relevant time frame, again, in that 15-minute exposure. So that, I don't know if we talked about that, but the drug is administered as a 15-minute exposure.
Yeah, and when we saw the promise of the preclinical data in both safety and tolerability and efficacy in biofilm models, in rabbit models, and the ex vivo, we then went to a phase 1 study and did a high dose intravenous infusion in healthy volunteers and found that we had no renal, hepatic, or cardiac side effects. It was safe and well tolerated in these patients at much higher levels than you would be exposed to with an irrigation or an injectable product locally. So because of the success and the ability to move forward after the phase 1 trial, we moved to a phase 1B, 14 patients using the product for knee, DARE procedures in PJI cases. And we had a low dose group and a high dose group, and we followed them for a year, and we saw that in the high dose group we had 0 reinfections and reoperations. In the low dose group we had 1 reinfection. So in a situation where at 1 year historical data would show that you would probably have about 50%, give or take, of reinfection, we had 1 out of 14. So that was very promising, and we've then aligned with the FDA, and we're now moving into our phase 2-3 pivotal trial that will be kicked off, hopefully enrolling a patient before the end of this year.
We'll definitely make sure to link all these studies in the show notes. Impressive work from your side. Now, speaking from a hospital economics perspective, antibiotic treatments for PJIs are inexpensive per dose, right? But extremely costly once you factor in revision surgeries, extended inpatient care, rehabilitation, etc. So how does 206 as a peptide-based therapy compare in terms of overall costs of care and value for hospitals? Because the value and the outcome for the patient, I think you already made that pretty clear.
Yeah, so I think the easiest way to think about it is, as Nick said, we're looking at this really as a paradigm shift. And what that looks like is, kind of for the hospitals or the surgeons, is for a given PJI patient, rather than the first thing you're doing is committing them to, kind of in the US, like 2 surgeries through a 2-stage revision process where you're going to have 1 surgery, take out the hardware, all of the associated care, like you mentioned, hospital stay, rehabilitation, et cetera. And then 12 weeks later, bring them back and do another surgery and pile those things on again. It's really what 206 allows us to do is 1 surgery, retain the hardware. And in general, you have lower need for kind of hospital stay. You eliminate a whole 1 of those, right? You have 1 surgery versus 2. But more, or as importantly, and the patient's able to stay mobile sooner, right? Because you haven't lost the joint. Longer term, you're helping to eliminate or reduce some of those comorbidities that arise because the patient is immobile for long periods of time.
If you think about, the impact that has, though, on all of the stakeholders, and that really intrigued me, was when you think about the patient, the surgeon, the hospital, the payer, and then even as you commercialize, what's the impact of that? The patient, just like anyone, you have a surgery, you think, it's going to resolve your problem. And if you get an infection, this is now a marriage sometimes to your physician in the clinic, and you're coming back over and over again. So the goal for a patient is 1 surgery, keep that device that you intended to solve your orthopedic problem and keep moving. For the surgeon, infection becomes a very high burden of care. These procedures take 2, 3, 4 times as long. Revision surgeries, infection surgeries take much longer than a primary hip or knee replacement. So you've got a tremendous burden of care in the operating room. These patients spend twice as long in the hospital. They come back to the clinic 5 times more frequently. And then, unfortunately, they have recurrence 25 to 50% of the time. So the surgeon is spending all this time and effort managing these patients, and what they're not doing is the opportunity cost of doing primary knees and primary hips and primary shoulders, which truly is a profit center for the hospital as well. They end up putting a tremendous amount of resources inpatient and outpatient, into taking care of these patients. So there's an opportunity cost, unfortunately, of treating infection patients where the hospital can never recoup the total investment of resources to take care of these patients. So the hospitals are losing money on infection cases. And then if you start to look at the whole patient journey from surgery number 1, 2, 3, home healthcare, antibiotics, skilled nursing facilities, multiple clinic visits, these are catastrophic economic burdens to, the charges going to a payer can be well over $500,000. So we really looked at the whole patient journey and tried to map that out of the impact across the board. And then to think of it as a medical device company, if you have a product like this in your portfolio, certainly you'll be able to treat and address the current customers you have, the surgeons in the hospitals that you have that you're already calling on. Say you sell total joint replacements, you can get into the operating room of the surgeons that you already have that use your portfolio of products. What's interesting when you have a product like 206 that is implant independent or implant agnostic, it now allows you to get into the operating room of all of your competitors as well, because you've got a new breakthrough therapy that is applicable to all total joint replacement, not simply those of your current clients are. So it's a door opener as well from a commercial perspective.
Impressive. Whenever a completely new therapy approach comes into clinical practice, patients and clinicians naturally wonder about safety. How do you address concerns regarding potential side effects, maybe based on your data of your last human studies? Did you even have dropouts? What kind of side effects did you observe? Or is this the next magic pill, so to speak?
Yeah, so we focused a lot on that, given the molecular class and some of the challenges that were associated with that. And so Nick mentioned one of the first things that we did was we tested in healthy humans with an intravenous administration to really understand if you got high levels of the drug systemically, what would manifest. And we didn't really see any issues, particularly in the kind of things that Nick mentioned around, renal issues, cardiac, et cetera. So the drug appears really safe when you administer it systemically. And then, obviously we did the animal studies where we would administer, very high concentrations of the drug locally in the joint spaces of, a couple different species, so rabbits and mini pigs. Didn't really see any significant issues there, that could be distinguished from kind of the surgical aspect of the trial, right? So you're doing surgery on the animals, you're cutting them open and you're kind of debriding as well. And so there was really no incremental damage to the tissues beyond what was seen in the controls. And then likewise, in the most recent human clinical study, we didn't really see anything that separated or kind of distinguished or was a signal above what typically happens in a patient undergoing DARE. So as of now, and we'll continue to follow these things obviously in the pivotal trial, but we don't see significant safety concerns in this patient population. And we did have a pretty, our patients in the phase 1B were pretty comorbid. So we had some folks with diabetes, kind of morbid obesity. And we tested it in a real world population and didn't really see any significant side effects, had nobody drop out because of the drug.
Yeah, and that's why we're going on, in our pivotal trial with a much larger sample size to continue, we hope to validate that safety and efficacy in a larger group that's randomized, controlled, going up against standard of care with a placebo control. So we're really, that's why you go and do larger studies to make sure that you can continue to demonstrate that safety and efficacy that we saw at least directionally in the early studies. And that's what we're doing next.
Definitely something to look out for in the future. Speaking of challenges and milestones, what challenges did you encounter in developing these technologies? And also, were there, maybe for you, Jonathan, key moments or milestones where you realized the impact of your innovation?
Yeah, I can answer that one. I think specifically, one of the big kind of overarching challenge is being viewed as an antibiotics company. And so just kind of this understanding that, that space kind of from an investment perspective and the development perspective has been pretty difficult. But I think more specifically, there were two specific challenges. So one was the molecular class. Like I said, it had been discovered. I think the first molecule was characterized in the 1980s. And in that subsequent time, nothing had really advanced because every time it got to a certain point in the development cycle, it fell out for safety concerns. And so a very common perception was that you just can't develop these kind of drugs for use in humans. And so
What kind of safety concerns? Sorry to interrupt you.
Oh, sure. Yeah, no, of course. A lot of times it's due to inflammation. If you look at the naturally occurring compounds, like, where you find these things in nature, they have two main purposes. So one is that they are immediately antibacterial. The second is that they are often inflammatory. So they call in the adaptive immune response. As you might imagine, like things that are purposefully inflammatory don't always make good drugs. And so that was part of our design, was to decouple those two things. So increase the activity, the antibacterial activity, but remove or essentially design out that inflammatory aspect. And so the early animal studies were very promising. But then when we did the human study, where we administered the drug intravenously, so high levels of systemic exposure, and saw none of those tox and safety signals, particularly around inflammation. That was a big deal, right? Understanding that, hey, these actually are developable. And then the second was something that's kind of followed along at the same time, which was this overarching kind of belief that if you have a device-related infection, the only thing you can do is cut it out. We don't have a way to cure the infection while leaving the device in place. And, again, the most clear example and kind of data that we've got around that was that phase 1b study where we treated 14 patients with PJI and, had that one recurrence versus the 50%, kind of that 6 to 8 range that you would typically expect.
Yeah, when you're developing something that's innovative and new and there's no predicate and you're going down a path, you're going to have a lot of people that will tell you it can't be done. This is really hard. No one's ever done it before. I don't have any analogs to look at. This is probably going to fail. It feels risky. And I guess I go back to the basics of, is there really a patient need? Are you hearing from patients? And we went and talked to patients. Are you talking to surgeons around the world? Is this a real problem? Is it, how unsolved is this? And we just heard time and time again that this is not only a devastating issue for total joint replacement patients, it has ripple effects across all the other medical devices that they have to deal with infection. And then, feasibility. Can you get through the benchtop, the animal testing, the human testing, and you're going to have hard problems along the way in development, in manufacturing, et cetera. Can it be developed? Can you get it through preclinical, clinical, and regulatory? Can you build a team that's wired in such a way that is willing to solve those hard problems, right? And that is not for everyone. And then finally, on the business side, is it investable? The market, ebbs and flows with what's investable. In the last 4 years have been challenging post-COVID. Can you secure investment? Is this something that can be reimbursed and be commercialized on a global scale? And can you continue, to demonstrate value, both clinically and economically, and for your shareholders as well, that you're really doing something that's going to make an exponential difference? And that's what we're really trying to do. But again, you've got to get it comes down to people. Can you get the right people to solve those really hard problems? And to me, once we saw the momentum from the preclinical work, it wasn't hard to find physicians and team members that wanted to be a part of this and help us tackle something really difficult.
And was there one key moment for you, Nick, that made you jump on the train and start pushing Peptilogics forward?
I've been in the industry for a long time. And I saw companies coming to me over and over again saying, look, we have this XYZ product that will decrease bacterial load on the surface of an implant. And they'll show the first picture where it's covered with bacteria. And then they'll show the second picture and 90% of the bacteria is gone, but there's still a lot of bacteria on that device. And they said, look, we've got this 2 or 3 log reduction and we think this is a great product. And I said, well, what happens to the bacteria that's still there? It's going to grow back. They said, well, yeah, but it removes most of it. And you start to think about, well, what are the regulatory implications if you can't resolve the infection? You can't get the claim that you're after. You can't then get the reimbursement you're after. So what I was looking for not only was something that had a tremendous clinical impact, it was an economic impact. And I'll tell you, when Jonathan called me, it was the fall of 2020, and he said we did the rabbit study, and no one had ever resolved this in this animal model. These rabbits just all died, and he said these animals made it, right, for the first time ever, and that was the signal to me. So I joined the company 2 months after that. We secured a $35 million Series B investment at that time. That was we saw in animals that we actually cured something that hadn't been done before in an animal model, and that was the trigger for me. It's one thing to remove bacteria on the surface of an implant. It's another thing to cure an infection.
Definitely. And a lot of people, especially startups, they are so driven by the technology, but do not think about the outcome in the end.
Yeah, and our outcome, is really we want to have patients that don't need another operation, that don't need to go back in the hospital, that don't need to be antibiotics for months or years. We want to resolve these things and cure infections. And that's why we're going after the data that we're after, the superiority claim that we're after, that's going to have an impact to be able to scale this and get it in every operating room in the world.
Yeah, and I think that's an important, you bring up an important point. Like it's not just enough to kind of technologically solve the problem. You have to understand and solve the entire problem. And that's one of the things we focused on very early, was understanding the disease from a clinical perspective, but also how that manifests kind of from an economic perspective. And really, as we developed the drug, it was really about kind of looking at all stakeholders, the way that they deal with the problem, and providing them the best solution for their particular problem. Like, so again, having it be surgically administered versus trying to do a typical drug thing, which is give a pill or stick it in an IV, where, as Nick said, there are real problems with trying to do that, right? It's not going to get there in the concentrations that you need to be able to clear the infection. So just being very thoughtful from the beginning across the whole kind of development cycle, from early lab data all the way through what the commercial product needs to look like to be successful. You've got to do that from the beginning.
How does Peptilogic approach research and development? And maybe you can also touch on your AI platform and how you identify those peptides and how you did it, and how you will also do it in the future to maintain a competitive edge.
Yeah, a lot of it stems from kind of how we identified 206. So like I said, it was we did a lot of work understanding kind of the basic science around kind of the class of molecules. And what that allowed us to do was develop a hypothesis for this idea of decoupling, kind of for this particular case, antibacterial activity from toxin safety concerns, or that inflammation aspect of it. Right. But if you take a slightly higher level view, that antibacterial activity and kind of lack of inflammation are really just drug-like properties, right? They're things that apply more broadly, kind of biologically, when you take any drug and put it into, the human body. And so we've been working on kind of more broadly understanding those things using some computational tools to, not just for this class of drugs, but just, again, kind of across the board, how do we link biological activity, whether it's toxin safety, blood-brain barrier penetration, things along those lines, to molecular sequence and structure, so that we can design molecules with the characteristics we want from the beginning. And so that's kind of at a high level how we do that. It's really about more of a design-focused approach based on what we did with 206 than it is kind of a typical, how do I say it, like a discovery-based, or kind of, just create giant libraries and screen through things to try to find something that works. It's more purposeful.
And the implications of being able to do those things and tackle those hard problems was enabled by, I guess, the approach we took with people. We knew that we were going to do something that most people would tell you can't be done. And you can't eradicate biofilm infection on a medical device without removing the medical device, right? You can't do it. You can't make a peptide safe. You can't have it be effective in an operating room. So you need a certain mindset. So our goal with the people in building our team, especially the R&D team, but it applies to every function in the company. Can we find the best people in the world, regardless of location? And that was a bit of actually a COVID advantage, because you weren't co-locating people. It wasn't necessary. It wasn't even possible. So when we were hiring our team back in 21 and 22, and even now, we focus on getting the best people in the world. We don't focus on putting them in the same office all the time. And it's the same thing with surgeons. We looked globally for surgeon and infectious disease input to get the best people in the world that have dealt with medical device infections, both in the clinic, in research trials, and have been frustrated with what they've been dealing with up until now. So we think we've got the best R&D team. I think we've got the best group of surgeons giving us feedback. And again, it's regardless of location. The goal is to get the best outcome and solve the hardest problem. So we've brought in both our internal team, our consultants and vendors, as well as our surgeon and infectious disease advisors that actually embrace solving really hard problems. And not everybody's wired to do that and take that risk. And to be able to forge a path going forward where you've got to write that story yourself, you're not copying someone else's or incrementally tweaking what someone else has done. And that, I think, getting the best people inside and outside the company is what's enabled us to solve those really hard problems. And we're really proud of the team that we've been able to build.
Yeah, I think I'd say kind of all the relationships. It's something that we focus on very specifically. As Nick mentioned, it's really about kind of identifying and working with kind of like-minded and the best partners. So this idea of kind of curiosity, creativity, and resilience, that applies across the board, whether it's from our investors, through our kind of manufacturing partners, through the surgeon relationships. It's really across the board the way we try to operate.
Yeah, it is needless to say that Peptilogic has been pretty successful, not only in your study outcomes. As you mentioned, you also survived the valley of death that appeared post-COVID. So could you discuss any key partnerships or collaborations that have been pivotal for your success?
Yeah, I mean, Jonathan hasn't been in a lab with 206 in years, right? So we've outsourced everything. And I mean, COVID also forced that, is that, we've outsourced for the last several years. So we've got great partners in CMC and manufacturing, in preclinical, regulatory, reimbursement, and our CRO that we've just brought in. We've had to bring in the best partners, but again, we've done this, you're right, with partners and collaborations all over the world. This is not something that we've done sort of in our own lab or in our manufacturing facility for the last 5 years. We've built this company, like I said, with the best internal staff and the best vendors and partners around the world, but we've done it, again, mostly virtual as a company.
Hire the right people. I think it's something as simple as that. I think one of the, we focus a lot on that, is making sure we find kind of folks who fit that mold that we've been talking about. But then I think it's also providing them the right types of problems in the right environment. So one of the things that we hear that's gratifying is kind of working on, these impossible problems. The types of folks that we're interested in, or that are successful with us, are the kind of people who gravitate to impossible problems and things that the conventional wisdom say you shouldn't do because that's not going to work. I know that's, to me, those things are really appealing. And I think it's most gratifying to watch people approach those problems that kind of really fires them up.
And Jonathan, coming back to something you mentioned, and it's a question to both of you. As a leader, how do you foster innovation and creativity? Can you maybe give advice to other young leaders in the field?
Yeah, we spend a lot of time before we hire a new wave of employees of what are the values, the attributes, the characteristics that we're looking for to ensure that sort of the phenotype of who we bring in helps us build on our vision. So we spend a lot of time thinking about who we want to bring in before we do it, whether it's an employee, a consultant, a vendor. And I think one of the things that we always talk about is we hire learners, people that are willing to learn and evolve their thinking and work around a difficult problem, that are willing to embrace risk and ambiguity. Not just, you hear these interview questions, how do you do under ambiguous situations? I want people to embrace that, kill something quick, be willing to fail in the short term to solve a bigger problem in the long term. So we encourage our staff to learn, to embrace risk, to fail fast, but not to give up on the problem, but to persevere and work their way around. And I always say we've got a great team of problem solvers.
And you mentioned that you traveled the untraveled road to some extent. So based on all what you've mentioned, what advice would you give to aspiring entrepreneurs? And Jonathan, your context maybe also to young researchers in the field.
Yeah, I mean, I think if you've thought really deeply about your problem and you're convicted in what you're doing, stick to it. I mean, it's sometimes that simple, right? It's an easy thing to say. It's much harder to put in practice. But I think you've spent a lot of time doing something You understand it probably better than anybody else, and just kind of stick to your convictions. That's the best advice I think I can give.
Yep. I always tell developers, to focus on, value-based development. Is there a real problem at the end that you're trying to solve? And when you do, it's going to have major clinical and economic impact that is easy for you to communicate. And if you know you've done the hard work, you've talked to the physicians and the patients, you have an understanding that you have a business case, and that you've probably talked to more people in the world than any investor, any partner you're going to have. If you know that you've done the hard work and you've validated that you've really got something with a clinical problem, it's feasible to get through the system, and there's an economic upside, have your conviction, stick to your conviction, and persevere, and don't let people tell you that it can't be done.
What are the long-term goals for your company, for Peptilogics, and where do you see Peptilogics in the next decade?
Yeah, so what we're really looking to build here is, and I think what we have an opportunity to build is, kind of a broader surgical therapeutics company. And I think, our goal isn't to kind of build this thing to sell. It's really about creating the next wave of company that can to kind of bring drug development and med tech together, right? Because I think one of the opportunities that we see is bringing kind of biology to a space that has traditionally been solved mechanically. And the implications of that, and I'll let Nick go into this a little bit more, is that if we do that ourselves, right? So we kind of go end to end where we have discovery through commercial, that unites two things that have been kept very separate in the past. And if we do that, it puts us in a really unique position going forward to create an entirely new category of medicine.
Yeah, if you think about, this is not just hard from developing, a new therapeutic, a new drug. It's also hard because of the model of which you have to do that, because it takes sort of a biotech pharma model economically of drug development. It's different than medical device. It costs more and it takes longer, and there's different kinds of risk, typically higher risk. But when the product gets through the door of the hospital into the operating room, now it's much more like a surgical product, a med tech. So we're a bit of biotech development, but med tech type commercialization, selling to people that do surgery and do procedures. So it's rare. You don't see your typical medical device manufacturers developing drugs and biologics. You don't see typical pharma companies commercializing things in operating rooms with surgeons and proceduralists. So we really think that the future, the vision, Peptilogics is really a surgical therapeutics company, able to develop therapeutics, but commercialize in a surgical environment. And that's rare. So we really see not only a very unique thing that we're developing in 206 and the implications for treating and preventing all medical device infections, but also building a very unique business model of being able to develop therapeutics, but commercialize to surgeons and proceduralists. And we think that's another bit of the secret sauce.
Last question. Where can our listeners learn more about your work?
Yeah, you can always check us out at the website, peptilogics.com. And we're pretty much attend all of the large kind of orthopedics conferences.
Yeah, we'll have staff.
So if you're ever there and you want to hit us up.
Yeah, we have staff at the Hip and Knee Society, both our internal team as well as our surgeon partners at the Hip and Knee Society, at American Academy of Orthopedic Surgeons, and as well as, the ATiO conference is particularly useful with your focus really on not just orthobiologics, but the technologies that address these musculoskeletal issues beyond the metal and plastic, the drugs and biologics that impact the entire musculoskeletal realm. I think, I've attended last year, and it was a tremendous meeting of the minds of academics, investors, commercialization partners to really think about what is the future of orthopedics and how are therapeutics, drugs and biologics going to impact across the musculoskeletal world. So I think the ATiO is a tremendous organization that's come to the fore in the last few years. And I think that's also a great place to learn about Peptilogics, but also to learn about all of the amazing technologies that are coming as part of the future of orthopedics.
Thanks a lot. It was a pleasure to have you on the podcast. And I'm also super excited to hear the update talk on Peptilogics next year at the ATiO Conference 2026. And congratulations again for winning the ATiO Best Emerging Company Award of 2025.
Thanks.
Perfect. Thank you.
We hope you've gained as much inspiration and knowledge from this conversation as we have. Remember, the journey of innovation and progress is an ongoing one, and the contributions of visionaries like our guests continue to shape a brighter future for us all. Stay tuned for more engaging interviews and discussions. In the meantime, check out our website at atioalliance.org for more information and content. Thank you for joining us.